Welcome, dear students. Today we have to discuss about how to control deterioration of crop varieties. Means today we have to discuss the steps involved in controlling the deterioration of crop varieties during seed multiplication or seed production program. So what are the steps involved, you can say, in seed production program for quality seed production to check the deterioration of the crop varieties? So one by one we will be discussing. The first one, use of approved seed only in seed multiplication. So here I want to clarify that if you are going to produce, whether you are breeder or whether you are seed producer or seed technologist or progressive seed grower, you have to obtain the approved seed. So if you are a breeder, you are willing to produce the breeder seed of your variety. What happens sometimes, if your institute has released a variety and its demand has increased and you are unable to meet the quantity of breeder seed required for whole country, so it may be given the quantity whatever you can produce at your institute, the institute which has developed the variety, that is okay. Apart from that, that quantity may be allocated to other universities or other institutions for breeder seed production program. So I mean to say that once you are going to produce the breeder seed, you must have the nucleus seed from the originating breeder first time at least. Then as you are breeder, you can produce nucleus seeds through SPS, single plant selection program or single panicle selection program. You at your own can produce the nucleus seed and the breeder seed. So I mean to say that you must obtain the approved seed for your seed production program. Okay, if you are an institution going to produce the foundation seed or certified seed, so for foundation seed and certified seed, you have to get it registered to your state seed certification agency. So they used to see that have you obtained the approved seed for producing your foundation seed. So for foundation seed, you must have the required quantity of breeder seed for your whole area for foundation seed production. So the seed certification agency will see the breeder seed tag, the how much breeder seed you have obtained, the approved breeder seed. Likewise in certified seed also, if you are a certified seed grower, whether you are institution or you are progressive farmer seed growers, then the seed certification agency has to see the tag, Have you obtained the approved foundation seed for producing the certified seed? So that is the approved seed. So it must be assured that you are using approved approved seed for producing your particular seed. Second, inspection and approval of field prior to planting. You have obtained the seed. It is now clear that you have got approved seed. Now your field has to be inspected by the seed certifying agency, state seed certification agency of any state. So the seed certification inspector has to visit your field once you have applied for producing the foundation seed or the registered seed. So the inspector has to visit your field, whether your field is as per the rules, is useful for producing the seed or not. So the major things they used to see that whether the location of the field is okay, that inspector or certifying agency officers may visit your field easily. That is one criteria for approving the field. You must have assured irrigation potential, because quality seed production cannot be done on natural rains. So you must have the field where the implements, the equipments can reach properly for sowing, harvesting and other things. Is your field is like that where the quality seed can be produced? Is it so fertile? So all those things are seen by the inspector and then they approve it, the field, prior to planting for quality seed production. Then come to next step. The crop has been sown. That also what inspectors see, that also they used to see that which crop and which variety was grown in that particular field. Because if the field was grown, say for example, last year it was grown by rice and the variety was maybe other variety, Swarna, and this year you are going to produce HUR 105, so they will not approve first, because there is chance of mixture. The more volunteer plants will be coming of Swarna, and you are going to grow HUR 105. So that has to be seen properly. Now come to next, after planting. You have planted. Field inspection and approval of growing crop at critical stages for genetic purity, detection of mixtures, weeds and noxious weeds and seed-borne diseases. Of course, this is the rule that seed inspector has to visit your field at different stages of crop growth for ascertaining that the variety which you are growing is genetically pure. He has the record, he has the characteristics, diagnostic characteristics of all the varieties being grown. So he will compare it, whether it is genetically pure when it is grown, not at only one time in growing period. He has to visit 2-3 times as per rule in different stages, in pre-flowering stage, in flowering stage, in maturity stages. So they have to see the genetic purity. Accordingly, inspector has to approve as per rule. Detection of mixtures, yeah. How much mixtures are there? Whether the variety which is being grown is the same variety. Whether the variety being grown having no mixture in the field while being grown. Weeds, there should not be much weeds. That should be in minimum standard seed rules. And noxious weed. Every crop has noxious weeds. Noxious weeds are those weeds, you can say, whose seeds separation from the crop's seed is very, very difficult. So they are put under noxious weeds. So that has to be seen very seriously. And seed-borne diseases. Yeah, inspector has to see that also. Because there are many diseases which are spreading through the seeds. So seed-borne diseases must be assured that is free from the seed-borne diseases. So these are the steps involved for quality seed production, for controlling the deterioration of the crop. Next, sampling and sealing of cleaned lots. Of course, the crop has been harvested, has been threshed, has been processed, grading has been done. Then you are going to take the sample. Means inspector has to visit the field for taking proper sample. There is a method of different topic it will be dealt. This sampling. So how to take the sample? The inspector has to take the representative sample, which is representing whole lot. Then sealing of cleaned lots. You have taken the sample, you are sealing it at your presence. Means inspector has to get it sealed at his presence. And then these seed samples are taken to the seed testing laboratories of state seed testing laboratories. There are many seed testing laboratories in the states. So these samples are being sent, and the laboratory tests are done for germination percentage, mixture, noxious weed, inert matter, all percentages are counted. So accordingly, the lot is being approved or rejected as a quality seed, depending upon the minimum seed standard rules. Then even if you are unable to, if the inspector is failing to judge the real purity of the seed while sampling, while testing in the laboratory, then he has to go for grow out test. He has to grow it. Grow out test I will be dealing in later lecture. Grow out test means he has to grow the sample of the seed along with the original authentic stock side by side, then you have to compare the growth and other things. For when sometimes dispute and confusion is there, then it is done, because it is very time taking. So that is done. So there are something I have to speak about this, the approval of field lots. Of course, here in inspection, field inspection during growing program, growing period, I could not explain about the inspector has to see the isolation distance also. Isolation means you can say, I'll be dealing this isolation in later lecture, of course, but little you must know that you are isolating means you are separating your seed field from all other field of same crop. Means you are separating your field, seed field, from field of other varieties of same crop. Why? To avoid cross pollination, unwanted pollination. If it is self pollinated crop, the distance will be lesser. It has in minimum seeds standard for every crop it has been described. We can see it later on. For every crop, different isolation distance is there. So for example, if it is rice self-pollinated crop, we are usually keeping 3 meter isolation distance from other varieties, from all the sides. Your seed field must be separated 3 meter at least minimum from other varieties of same crop, from all the sides, so that you know that about 5% cross pollination is occurring in self-pollinated crops too, which you are not visualizing it. So it has been calculated that how much for purity, maintaining the purity, how much which crop you can keep, how much distance. So distance is there for maize. If it is foundation seed, if it is breeder seed, for everything is different. For maize, 400 meter minimum distance, because it travels, your pollen grains travels in the air, lot of distance. So the pollen grain of other varieties should not come in your seed field. So this is the isolation distance. And in isolation, I will be dealing later on. Of course, little I want to speak here also. So whether you are maintaining this distance, if not, say for example, all farmers are growing their own varieties, different varieties, how you are going to keep? If it is not possible, or maize 400 meters is too much, or pigeon pea 200 meters, it is too much. So is it possible? So what has to be done? So if it is maize, or for every crop, the time isolation is also there. If farmers have grown nearby your field, other variety, so wait for some time. It is depending upon crop to crop, 21 days to 30 days difference if you are making. So this is time isolation. So you are checking, the other farmers have grown it, you have awaited for certain period, 21 days, 30 days, depending upon the crops. So flowering in the field of those farmers are coming earlier and they are finishing, then your flowering is starting. So there is no chance of coinciding the pollination between these different varieties. So time isolation is there. So if it is not possible, then mechanical isolation is there. So in many crops like rice, in hybrid rice cases, particularly if you are not able to have that much distance in hybrid rice seed production program, because you are using male sterile lines, so you minimum 100-meter distance, 200-meter distance are required. So if you are not having that much, so can you put metals to all the sides? That is called mechanical isolation. Or even you can use that what you call for isolation this live plants, like in case of rice we are using Sesbania, 5 rows, 10 rows of Sesbania to all the sides of your seed field, hybrid rice seed production field. If you are not able to have 100 meters or 200 meters distance from other fields, so we can use that live fencing with Sesbania like crop, 5 rows, 10 rows, whatever possible from all the sides, so that you can check the pollen grains of other varieties coming to your field. So these are the ways how you can check the cross pollination in your field, the isolation. So that is being seen by the seed inspector while field inspection and approval of growing crops. So that is also seen. And also, if you say that here the seed for foundation seed and certified seed, seed inspectors are coming, but for breeder seed production, because this is being produced by the breeders of different institutions, particularly the ICR institutions and SAUs, State Agriculture Universities or Central Agriculture Universities. Because centrally, the breeder seed production program is governed by centrally governed. By what? National Seed Project of ICR. I have explained in my other videos. It is governed by the National Seed Project of ICAR. So the ICR is allocating to different universities this much breeder seed of this variety. You have to grow, you have to grow, you have to grow. And at the time of growing period, as here you are seeing that in foundation seed and certified seed, seed inspectors are coming and visiting the field. And in breeder seed production program, you are a breeder, you are a responsible person. It is hoped that you are doing all the things at your own. But of course, at least once in a crop period, at proper critical stage, particularly it is seen that at the time of heading or at the time of maturity or grain filling stages, the monitoring team of ICR visits throughout the country. For many centers, different monitoring teams are made by the ICR. In annual meeting, it is decided that which team is given which are the centers of different states. So they are usually controlling the quality of breeder seed production program. Here in foundation seed and certified seed, the certifying agency is taking care of that. So these are the things. Then sampling and sealing, and I have discussed this also, growing also this. So I think I have covered the steps involved in controlling the deterioration of crop varieties, means producing the quality seed. These are the steps have to be followed. So thanking you very much for today. I'll be coming next with the next topic. Thanking you very much again.